Hi everyone, I'm excited to talk about topological cluster statistics, which is a novel brain inference approach that uses information from structural connectivity to derive anatomically informed brain inferences. Today's talk will include an introduction to inference for brain imaging. We'll then talk about anatomical brain wiring maps, and we then try to link the two and explain what topological cluster statistics does. Finally, we'll end with some concluding remarks and points for future directions. So to discuss inference for brain imaging, we need to talk about statistical parameter maps. You're seeing one on the screen right now. On top, we see surface projections of the cerebral cortex, and on the bottom, we see similar projections for subcortical as well as cerebellar brain regions. Essentially, a statistical parameter map is a set of statistics reported for any point on the human brain. The statistics could be p-values, t-statistics, or Cohen's d effect sizes. Regardless of what statistics we report, the problem at hand is the fact that we are running multiple tests, essentially hundreds of thousands of tests at every single point of the brain. Now this brings about the problem with multiple comparisons, and the fact that if we don't correct for the multiple statistical tests that we are running, we're going to run into the problem of having inflated false positives. Basically, if we use an alpha of 0.05, 5% of brain regions are going to be inferred as significant differences just out of chance. And to show you how severe 5% of brain regions would look like, this yellow region is just 5% of the whole brain. So it shows us how important it is to correct for multiple testing. Now there are different approaches that could be used for correcting multiple tests. There are some general statistical approaches that can be used for this purpose, such as the Bonferroni correction for the family-wise error rate, or the Benjamini-Hochberg correction for false discovery rate. However, the fact is that when using neuroimaging data, these general statistical correction methods are usually not powerful enough in that they don't capture most of what we are interested to capture. That's why we use MRI-specific approaches to correct for multiple testing. One of the most commonly used MRI-specific approaches is the cluster statistic. The idea behind cluster statistics is that it is more likely for a region to be activated or deactivated together, and therefore a cluster of activity is less likely to be noise and more likely to be an actual signal to be inferred. Now the question that we are having is whether this idea could be extended to anatomical information of the brain. We know that the human brain is a complex network of many neuronal connections connecting different regions of the gray matter together. And the best way to capture these connections in a living human being is using diffusion-weighted MRI and running tractography algorithms. We have recently developed a method to map high-resolution brain networks using tractography. Now, the idea behind TCS is to use this anatomical connectivity information as a guide for inference in a cluster inference paradigm. Let me explain the idea behind TCS using this simple example. We simplify the brain in a two-dimensional ribbon, and different parts of these ribbon indicate different brain regions. Let's say we have a statistical parameter map that looks something like this. We have three regions of activity. Two of them are a bit bigger, and the third region, shown on the right-hand side, is a bit smaller. Ideally, we want an inference approach to detect the three green regions that are true positives in the statistical parameter map as inferred clusters and ignore the rest as noise. So a traditional cluster inference approach would count the number of significant pixels within each cluster, and that would indicate the likelihood of that being a significant result. So we can see that two larger regions of activity would be detected as significant. However, this approach would miss the smaller region of activity and ignore it as noise. The idea behind TCS is if we had information about the underlying anatomical wiring of the brain, we could use that information to reconstruct the network that was implicated in that activity pattern, and therefore we would ideally be able to detect not only the larger regions of activity, but also that smaller region. We would also be able to provide an information about the implicated subnetwork that constructed that activity pattern, and this could be useful for interpretation of the findings. So the big question is whether this simplified example also applies to real-world data. That's why we use data from the Human Connectome projects. We have used 5 different task contrasts, and we have run a wide range of power assessments on sample size, cluster defining thresholds, and evaluated sensitivity and specificity of TCS. Instead of reporting all these detailed analyses, for the purpose of this talk, I will focus on a single task and show you what TCS does compared to traditional approaches. For the purpose of this talk, let's focus on the emotion tasks and the faces versus shapes contrast. We want to see how TCS performs compared to traditional cluster statistics. We first built a putative ground truth inference by running Bonferroni correction on the observed effect in the whole population of 1,000 individuals. The question is how much of it we can detect in a limited sample size. So if we only had a limited sample of 40 individuals, how much of the ground truth can we capture with the traditional and the new approach? This picture shows the brain regions that pass the cluster forming threshold. We can see that there are some regions that are the true positives and we want to detect them, as well as regions that are false positives. This picture shows the effects that remain after thresholding the statistical parameter map. Ideally, the inference approach would detect all green regions and ignore the false positives. So these are the results from a traditional spatial clustering approach. We can see that all large regions of activity are detected, however, smaller regions are dismissed using this method. More accurately, this method only tells us about the clusters, and it doesn't tell us anything about the connectivity patterns between these clusters and the potential anatomical reason that these regions are activated or deactivated together. So this is what the results from TCS would look like. We can see that TCS detects all those larger regions of activity, but also it detects smaller regions of activity that was missed by the previous technique. Interestingly, TCS also provides information about how these regions are interconnected. Looking at this data, we can see that the majority of inferred significant differences form a single network of interconnected regions spanning the whole brain. Now, given that we have information about this network, we could use different tools from network neuroscience to analyze this pattern of activity. For example, we can project the information about the implicated anatomical network linking these regions of activity onto a brain atlas. This can tell us that there are different structural hubs that support this particular activity pattern. We can see that regions of the visual and attention networks of the brain form the hubs of this particular task. Alternatively, we could project the same information into a 3D visualization of the brain networks to see how these different regions of inferred activity are interlinked. An interesting interpretation that we can have from this format of visualization is how the 2 different hubs of the attention and visual networks are connected through a relay station in the amygdala. Basically, the amygdala forms a bridge between the visual and attention networks when performing this particular task. We have additionally performed a range of alternative analyses to show how TCS can enhance the power and sensitivity of inference by using anatomical information. However, for the purpose of this talk, I won't go into the detail of these analyses. So I would like to leave you by a few concluding remarks. We presented a novel inference approach, the Topological Cluster Statistic. This approach uses structural connectivity information to enhance traditional cluster-based inference methods. Not only does TCS improve the sensitivity of the statistical inference, It also provides us with network information that can aid for data-driven interpretation of the activity patterns. I should mention that TCS is being made publicly available as part of FSL PALM, so it can be easily integrated into your current pipelines. If you'd like to have a closer look at our findings and have a read at our preprint, make sure to check out this link to the Git repository. I would like to end today's talk by acknowledging that the results presented today have been a team effort, and I thank all my colleagues and co-authors who have helped in the development of this method. And also thank you for listening.